I apologize for my abrupt departure yesterday. If you've read anything in the news about people getting their identity stolen during the time when everybody's spending more time online, that happened to me not long ago. So since then, whoever has my identity has applied for unemployment in my name. They've bought a house in Texas. They've smuggled drugs across the border. They've shot a few people. They've opened bank accounts. So I've filed identity theft notices with the government, and that was the government calling me back to ask some questions. And unfortunately, when you call them, they're always busy, and they tell you that if you don't answer the phone when they call you, that they're going to take you off their list, and you have to call them again and get back in the line, and they'll call you again whenever they're ready. So I kind of had to talk to them when they called, but I apologize for having to leave class so quickly. We thought my wife could talk to them at first, but that turned out to not work so well. Are there any questions about what we talked about yesterday before we stopped so abruptly? If not, I'm going to go back to that. And keep in mind, right now we're running about probably close to a week, not exactly a week, not quite a week, but we're running a little bit behind schedule. So tomorrow we'll be talking about the first short paper you're going to write. And I know sometimes students, when they panic when they hear they're going to write a paper, but keep in mind our paper really is going to be short. It's going to be 1 to 2 pages. It's not going to be longer than that. And when we write our papers, we're going to write them on various groups of organisms that we're talking about. So we're just about through with evolution. And when we finish evolution, we're going to start on groups of organisms with prokaryotic cells. So we're going to be writing a brief report about one of those groups. I'll give you some more details tomorrow. So I think we were still looking at the geologic column when I left so quickly the other day. So keep in mind what I said. When we look at the geologic column, the oldest time periods are typically at the bottom. The youngest time periods, the most recent times, are towards the top. Now, if you would be looking at rocks in some particular location in the world, keep in mind that usually all these time periods are not represented in the same area. We have to go from region to region, and we have to kind of piece together the overall period because the way that these rocks are deposited, they're only deposited under certain geological conditions. Now you might think, okay, well, if we find some rocks here in Illinois and we don't have the complete geologic time scale, how do we piece things together? If we look at the minerals in the rocks and things like that, there are actually consistent layers in the rocks. And so we can look at a layer from here in Illinois. We can compare it with a layer that we discover out in Colorado. We can compare it with a layer that we find in Kenya. And we can line things up so that if we have some layers in the rocks in Illinois, some layers in the rocks in Colorado, some layers in the rocks in Kenya, we can fit the different pieces together. For instance, if we look at this time that's designated as about 65.5 million years ago between the green and the yellow. This is when dinosaurs went extinct. Right now, the evidence is very strong that one of the major factors that caused the extinction of the dinosaurs was that an asteroid hit the Earth. And one of the pieces of evidence for an asteroid hitting the Earth is if we look at the rocks at this period of time, there's one particular element called iridium found in the rocks. And iridium is a very rare element on Earth, but it's much more common in asteroids. So we think what happened was when the asteroid hit and there was a giant explosion, that this iridium from the asteroid was kind of pulverized and sent out. And even though this asteroid probably hit near the Yucatan Peninsula in Mexico, we can find an iridium layer in rocks in Siberia and in Australia. and areas that are quite distant. And the types of rocks that we find in those areas are similar to the types of rocks that we find in the Yucatan area, where this iridium was first discovered. So there are some geological clues that we can use to kind of fill in our gaps. So we want to keep that in mind. Now, when we look at the various time periods, notice if we go down further to this gap between the kind of bluish and the green, there's the Permian-Triassic extinction event. Most people are familiar with the fact that at some point in history, most of the dinosaurs became extinct. But what you may not know is there are a number of minor extinctions in the history of living organisms on the Earth. And there are 5 mass extinctions that are bigger than the other ones. Now notice if we look at these 5 mass extinctions, in all of these extinctions, at least 70% of the groups of organisms on Earth have gone extinct. Remember in our classification scheme earlier, that we have genus and species. Whoops, that's on the next sheet, sorry. We have genus and species, and above the level of genus, there's a family. So usually we think of how many families of organisms went extinct. So in this case, what we see is that up to 90% of the species have gone extinct in a mass extinction. Now keep in mind, there are always extinctions happening, and extinctions occur when the last living organism in a group dies. This is called the background extinction rate, but a mass extinction occurs when a lot of living organisms become extinct in a short period of time. So if we go back and we look at the KT, which is the Cretaceous Tertiary extinction event, C is already used as the abbreviation for another geological time period. So Cretaceous gets K. If we look at that, even though it says 65.5 million years ago, it really stretched from about 66 million years ago to 64 million years ago. Dinosaurs didn't become extinct overnight at one instant in time. So we want to keep that in mind. So most biologists think that if we look at what's happening today, that right now there's a 6th major mass extinction occurring. And this one is different than the earlier ones. If we look at likely causes of the earlier mass extinctions, typically they were due to changes in global climate, either global cooling or global warming. They were due to asteroid impacts. They were due to volcanic activity. And we're not talking about 1 or 2 volcanoes occasionally erupting. That still happens today. We're talking about volcanoes erupting over large portions of the Earth. Today, the mass extinction is occurring because of human activity. We're destroying the habitats that living organisms survive in. We're changing climate. We're polluting various parts of the environment. And all of these things contribute to extinction. But the extinction rate today is about 1,000 times higher than the normal background extinction rate that's always happening all the time, where an organism goes extinct now and then. Now, when we think about living organisms, one way to think about this is to think about this kind of like a business. You know, suppose Ford and Chrysler and Volkswagen and Toyota and Honda and Kia and all the companies that make cars suddenly went out of business. There would be a huge business opportunity for new companies to start building cars. And this is one of the things that we see after a mass extinction. We typically see something called and adaptive radiation. One way to think about adaptive radiation is to go back a few days ago when we talked about allopatric speciation. And remember, in allopatric speciation, the first step was geographic isolation. And then the second step was divergent evolution. The two populations that have split apart become different. Now, keep in mind what we said the other day. There's some argument about whether this divergent evolution happens very slowly and gradually, gradualism, or whether it happens relatively quickly, punctuated equilibrium. But when we see adaptive radiation, we see one species that diverges into two, and then those two species may diverge. And so we have something that looks like a lot of crisscrosses on our phyletic tree. We'll look at an example in a little bit. But this is what happened after the KT extinction event. If you notice, the first mammals appear down here at the bottom of the green, about 240 million years ago. And if we look at the mammals up here that are at the top of the green, the mammals that are alive are very small animals. The big mammals are maybe the size of dogs, but most mammals are small animals. They live in burrows in the ground. They're mostly nocturnal. They come out at night. The dominant animals are dinosaurs. So one of the mistaken views that some people have is that dinosaurs went extinct because mammals evolved, and then mammals were so much smarter and so much better that we drove dinosaurs into extinction. But that's not what happened at all. Actually, if you look, the first mammals appear down here, and dinosaurs don't really evolve until a little bit later. So mammals were around at the same time as dinosaurs, but the dinosaurs were the dominant life forms. Mammals started to go through an adaptive radiation after the dinosaurs became extinct. So did birds. So when we look at an adaptive radiation, there's kind of an open environmental opportunity. You know, when the dinosaurs went extinct, big dinosaurs like Tyrannosaurus and other predators weren't there anymore. So now, in a sense, there was a business opportunity for large predators, for large carnivores. And mammals started to fill these vacancies, if you will. So that's one way that we get adaptive radiation, is after a mass extinction. All of a sudden, there are a lot of opportunities in the environment. And keep in mind, one of my favorite things to complain about, students who had me for 105 know that, you know, when we talk about large predators, if you've watched the Jurassic Park movies, most velociraptors were about the size of turkeys, but they're not very scary if they're as big as turkeys. So the movies had to exaggerate a little bit. Now, we do see adaptive radiation in another situation. And a good example of this is if we look at a type of plant called Hawaiian silver swords. And Hawaiian silver swords, we have pretty good evidence, came to Hawaii about 6 million years ago. And the way that we have good evidence is through studying fossils of older, dead plants. And they probably came to Hawaii from British Columbia or maybe Washington or California State in the United States. But when they got to Hawaii at the time, Hawaii was not the lush tropical rainforest that we necessarily might see today. The Hawaiian Islands are volcanic islands, and there wasn't a lot of plant life. So as the seeds for the original silver sword plants spread, what we see today is we see silver sword plants that kind of are large cone-shaped structures that look a little bit like a cactus. We see silver sword plants that are small and look like kind of moss. We see silver sword plants that look like big spongy balls. We see silver sword plants that are trees and bushes. In fact, if you look in the Hawaiian Islands today, there are about 28 different species of plants that live in very different environments and survive differently that we think are all descended from the silver swords that originally, we don't know if a bird carried the seeds to Hawaii or if they floated out on the water. We're not sure how they got there. So this is another way that we can see adaptive radiation, not necessarily after a mass extinction, but if there's an ecological opportunity that really isn't occupied. And when these plants got to Hawaii, there was an ecological opportunity. There weren't a lot of plants there. So there was an opportunity for them to go through divergent evolution over and over again. We see the same thing if we look at fruit flies in the Hawaiian Islands. You've probably all seen fruit flies in your kitchen or something like that. There are hundreds of different species in the Hawaiian Islands. And these species arose because the islands are kind of separated from each other. You may not be aware, but there are some fairly high mountains on the Hawaiian Islands. So they tend to block the fruit flies from going to the other side. We see the same thing in a much broader sense when we look at insects. Insects were some of the first living organisms to colonize the land. So when insects got to the land, there wasn't really much competition. And probably because they were one of the first groups, today there are about 3 million species that scientists have studied and named and classified. And out of those 3 million species, about 1 million of them are insects, about a third of them. Most people think probably insects make up, more than half of all the species of animals on the earth, maybe more than half of all the species, period, even counting plants and fungi and other types of living things. So when we look at adaptive radiation, we can't see adaptive radiation happening. What we can see if we look at all these silver sword plants is we can see kind of what the result of that adaptive radiation is. But even though this may have happened by punctuated equilibrium, remember what we said, we're still talking about hundreds or more likely thousands of years to get a new species through punctuated equilibrium. So we're not talking about something that can typically be observed in a human lifetime. We can see bacteria form new species in a human lifetime. Beyond that, we have to be pretty lucky and be in the right place at the right time. So what we want to keep in mind is all these processes: adaptive radiation, mass extinctions. Go back to some of the ones we were talking about yesterday: Mullerian mimicry, Batesian mimicry, coevolution between pollinators and the flowers they pollinate. These are all different patterns that we can only observe by looking at more than one species at a time. So we can't really study these patterns that we see in macroevolution through just looking at a gene pool, because a gene pool is just the alleles for one type of living organism. And these all involve more than one organism. So are there any questions about these basic patterns that we see? And notice there are some others that are not quite as common, but that we do see. Okay, if there aren't any questions, we're going to go to a slightly different topic. Because we've been talking about living organisms. Can you see the page that's titled Systematics, Taxonomy, and Phylogeny now? Or can you not see it? Let me ask that.
I don't see it.
Don't see it?
I see it.
Okay, let's go back and let's see if you can see it. Can you see it now?
Yes.
Okay, So let's remind ourselves how at least some living organisms are classified. If we look at plants and animals and fungi, they're all classified in the domain Eukarya. And then whether they're a plant or an animal or a fungus, that's their kingdom. And then the kingdoms are subdivided into phylums. The phylums are subdivided into classes. The classes are subdivided into orders. Orders are subdivided into families. Families are subdivided into genera, and finally, genera are subdivided into species. Now, remember what we said? If we look at most prokaryotic organisms, or if we look at other eukaryotic organisms, when I say other eukaryotic organisms, the ones that aren't plants or animals or fungi, the ones that are protists, what we see is that those organisms, usually we just talk about their domain and their genus and species. Some people do try to use kingdoms and phylums and classes with them, but the people who do that, there's not very much agreement on how those organisms should be named. So we'll talk about that when we look at each of those groups. Well, this system of classification, and classification is a science called taxonomy. This began back in 1735. And 1735 marked the publication of a book called Systema Naturae, the System of Nature. And in Systema Naturae, the author, who was named, well, kind of was named Carolus Linnaeus, proposed how living organisms should be classified. Now, the reason I say he was kind of named Carolus Linnaeus, his name was actually Carl von Linné. But at the time, think about 1735, Europe was coming out of the medieval time period, which is sometimes called the Dark Ages. And during medieval times, people who were well educated could read and write Latin. And so Carl von Linné, like a lot of people, he wasn't the only one, Carl von Linné gave himself a Latin name. He named himself Carolus Linnaeus, which wasn't his real name, but was kind of a Latin version of his name. And the reason he did it was because educated people knew Latin. So a lot of people gave themselves Latin names so that they thought it sounded more impressive. So think of it like today, a lot of entertainers give themselves, you know, a different name. They don't use their real name. That would be too boring. Now, before Linnaeus, biologists had named organisms using something called a polynomial naming system. And remember from back in 105 when we talked about polysaccharides and polypeptides and polymers, poly means many. So a polynomial naming system uses a lot of names, many names. So the common European honeybee was something called Apis pubescens. The problem was when the common European honeybee was first named, Apis pubescens was okay. Everybody knew what that was. And this was true for most species that Europeans encountered on a regular basis. But this started to change after Columbus's voyages and other explorers started going around the world. Because if you looked at the European honeybee, it was pretty much the same across all of Europe. So Apis pubescens was okay. This was a good way to describe it. The problem was when Europeans got to North America, there was a different type of honeybee. And when they got to Africa, there were still different honeybees. And when they got to South America, there were still different honeybees. So what they started doing was to name honeybees, and I'm focusing on the European honeybee now, but we could talk about the North American honeybee or the South American honeybee or the African honeybee. It would be similar. They started giving it a series of descriptions. So they called it Thorace subgrisco, which means it has kind of a gray thorax. The thorax is the chest area. Abdomini fuso, which means the abdomen is fused to the thorax. Pedibus posticus, Ped is foot, post is backwards. So the foot kind of looks like it's pointing backwards. So by the time Linnaeus came around, if you wanted to talk to another honeybee, if you wanted to talk to another scientist about the honeybee, to make sure that you were both talking about the same honeybee, you had to use this whole long name starting with Apis pubescens. Because if you didn't use all of this, maybe you were talking about the North American honeybee and I was talking about the South American honeybee. And Linnaeus proposed a system of what he called binomial nomenclature. And binomial refers to two. And he said that things could be named by using a genus and species. So the European honeybee became Apis mellifera. Remember what we said earlier in the semester. If we look at genus and species, the genus is capitalized, the species isn't, but in print, they're italicized. In handwriting, they would be underlined. Well, how could you tell which honeybee it was? And this was because Linnaeus introduced the taxonomic or classification levels of families, orders, classes, phyla, divisions, and kingdoms. Notice that he didn't use domains. That's relatively new because that's based on the cell type. And if you go back to Linnaeus's time in 1735, nobody was really looking at cells as being prokaryotic or eukaryotic. Now today, we can talk about plants, animals, fungi. Some people say bacteria are a separate kingdom. Some people say archaea are a separate kingdom. But notice in Linnaeus's time, there were only two kingdoms. Everything was either a plant or an animal. Things like mushroom, which are considered a fungus today, mushroom was considered to be a plant. It didn't really move. Whether they could move or not was one of the main things that separated plants from animals. So let's look at the classification of the American black bear, just as an example. So if we look at the domain, which is shown on the far right, American black bears are in the domain Eukarya. And eukaryotes have eukaryotic cells. So they have a cell with a nucleus and membrane-bound organelles. And so this shows some examples of eukarya. Since we're going to mostly be talking about mammals because bears are a mammal, they show a few fish and they show some plants, a few other things, but they mostly show mammals. That's just because they're going to use the same pictures all the way through. But don't make the mistake of thinking that most animals are mammals. Most animals, think about what we said a minute ago, are insects. Now, one of the kingdoms that's in the domain Eukarya is Animalia, the animals. And this includes sponges, insects, like that looks like a monarch butterfly. I think that's a starfish, a fish, a bird, frogs, zebras. So these organisms are all Animalia. Well, eukarya all have eukaryotic cells. So notice Animalia is a subdivision of eukarya. So all Animalia, all animals have eukaryotic cells. But they also have some other characteristics. I mentioned before, Linnaeus, when he thought of animals, thought about things that could move. However, sponges don't really move much. But there are some technical things that we can use to define an animal. Animals developed from a stage called the blastula. The blastula is one of the very early stages after the sperm fertilizes the egg. In a person, the blastula is what makes chemicals that tell the mother's uterus to start building a placenta. And the blastula, these chemicals it produces, this is actually what we test for in early pregnancy tests. Now, if we look at animals, there are a lot of phylums in animals. There's the phylum Porifera, which is the sponges. There's the phylum Arthropoda, things that have jointed legs, that includes insects. One phylum in the animals is the chordata. Now notice, chordata are a subdivision of animals. So that means chordata developed from a blastula, like animals, other animals, and chordata still have eukaryotic cells. So as the taxonomic categories get smaller and smaller and smaller, they always have the characteristics of the larger category. Well, chordata have a stiff rod that runs along their back. It's actually called a notochord. And chordata includes a couple of groups of fish, birds, amphibians like frogs and salamanders. They don't really show any reptiles, but it includes reptiles, and it includes mammals. Now notice, in people, even though we're in chordata, the notochord, the stiff rod along our back, that actually disappears very early in our embryonic development. And the notochord gets replaced by what we call our vertebrae or our backbone. Now, within the chordata, there are a lot of classes. There's a class that includes sharks. There's a class that includes most of the other types of fish. There's the class that includes amphibians, the class that includes reptiles, the class that includes birds. Bears are classified in the class Mammalia. And there are some technical definitions of mammals having to do with how many holes there are in the skull back behind the ear. And these holes are places where blood vessels and nerves pass through the skull from the inside to the outside. But the definition that most of us learned back in grade school is still pretty good for identifying mammals. Mammals have hair and they suckle their young with milk. Now notice, some mammals like whales have hardly any hair. And if we look at people, people only have hair in a few locations. On the other hand, some mammals, if you have a dog or a cat, or if you've seen a horse, they have hair all over their body. Now, if we look at mammals, since they're chordates, they have a notochord during part of their life. Since they're animals, they develop from a blastula. Since they're eukarya, they have eukaryotic cells. So again, we want to keep in mind, every level always shows the characteristics of the levels that, in a sense, contain it. Now, there are a lot of groups of mammals. There's the rodentia, the rodents. There's the primata, the primates, the cetacea, whales and dolphins. Bears are in a group called carnivora, which includes things that we think of as bears, things that we think of as cats, things that we think of as dogs and wolves. And if we look at carnivora have certain characteristics of their teeth that typically identify them as being carnivores. Not to mention the fact that when we look at carnivora, they eat other animals. That's what a carnivore is. Now, keep in mind, some carnivores, and black bears are actually an example. Black bears will also eat berries. So they don't only eat other animals, but at least part of their diet comes from other animals. Now, within the carnivora, there are different families. There's the Phylogeny, the family that includes cat-like organisms. The Canidae, the thing that includes wolves and dog-like organisms. Bears are classified in the group Ursidae, which is bears. Notice you can tell from the diagram, this also includes pandas. You might not realize it, but pandas are bears. One way to remember that Ursidae is bears is, if you know anything about astronomy, remember the Big Dipper and the Little Dipper are called Ursa Major and Ursa Minor. So Ursa is from the same root word. Now, within Ursidae, there are several different genuses, or the plural is genera. Bears are in the genus Ursa. Now notice Ursus is italicized here. Remember, genus names are italicized and it's capitalized. And then the black bear, the American black bear, I should say, because there are other types of black bears, is Ursus americanus. And notice the species name always starts with a lowercase letter. And remember, I think I told you earlier, we never list the species name by itself. We always either write out the genus name, or since we just mentioned Ursus, we could write U period americanus, but we never just say americanus. And this is because the same species name might be used for American black bears, but it might be used for other living organisms that are found in America. So if you just say americanus, we're not sure what you're talking about. But if you say Ursus americanus, this is the only organism that has its name. Now, one of the things that we have to keep in mind when we're talking about a species, and black bears are an example, because black bears are found across really all of North America and some of Central America. If we look at black bears, right now, all black bears are considered to be one species, Ursus americanus. But there have been times in the past where black bears have been subdivided into 2 or 3 different species. And remember, one of the things that we said when we were talking about species the other day is if we have populations that live in very different areas, if you have a population of black bears in Georgia and a population of black bears in Canada, they don't really mix and mate together. So it's hard to tell if they're one species or not. So one of the things we see in classification is there tend to be kind of 2 opposing schools of thought. Some people who classify living organisms are what we sometimes call lumpers. So they look at all the black bears across the Americas and they say, yeah, they're all basically the same. They're all one species. And some people who classify organisms are what we call splitters. And they look at the black bears and they say, well, you know, if we look at the population of black bears in Montana, they have some different characteristics than the population of black bears in Michigan. So they're really different species. Who's right and who's wrong? I wish I could tell you, but I have no clue. This is something that happens not just with bears, but with all sorts of living organisms. There are people who are splitters, who divide them up into several species. And there are people who are lumpers, who group things together and say, yeah, they're all basically the same. So when we think about this, we want to keep in mind that, you know, there are different ways of looking at this. Even with animals, where our basic definition is, remember, a group of populations, and the individual members of these populations can interbreed. In other words, they can reproduce together, and they can't reproduce with members of other populations. Even though that might seem like that's an airtight definition, there's still room for argument. You know, if organisms can interbreed, but if they live in very different areas, so they never meet, they never do interbreed, is it really correct to call them members of the same species? That's something that's more a philosophical question than a scientific question. And so, you know, every 10 or 20 years, the splitters become dominant, and then they subdivide species into many species. And then the lumpers become dominant 30 years later, and they group the similar organisms together into one species. So keep that in mind. If you would look something up, you might see one reference that says, oh, there's one species of American black bear. You might see another reference that says, oh, there's 4. Doesn't mean that's one is wrong and one is right. Different ways of looking at the problem. So when we look at classification, even though it seems like, you know, this is more than enough subdivisions, because the domains contain multiple kingdoms. The kingdoms contain multiple phyla. But when we look at living organisms, what we see is a lot of times, whether people are lumpers or splitters, one of the things that's happened is people have added intermediate levels. So for instance, when people talk about kingdoms, they sometimes talk about subkingdoms. Now, when you see a subkingdom, think of like a submarine. Sub means under. Submarine goes under, marine is water. A subkingdom is under the level of a kingdom. So sometimes animals are divided into two subkingdoms. Kingdoms or subkingdoms, if we have one, can be divided into phyla. And a phylum can be divided into subphyla. So for instance, if we go back to our bear for a minute, if we were talking about people, people are also in the domain Eukarya, the kingdom Animalia, the phylum Chordata, but the phylum Chordata is divided into several subphyla. We're in the subphylum Vertebrata, meaning we don't just have a notochord, we have vertebrae. We have a skeleton made of bone. So sometimes we have subkingdoms. Now, when we get to classes, we sometimes see something called a superclass. Super means above. So a superclass is above the level of a class. It's below the level of a phylum or a subphylum still, but it's above the level of a class. So we can have a superclass, a subclass, we can have a superorder, an order, a suborder, we can have a superfamily, a family, a subfamily. With the genus level, we don't usually talk about a supergenus, but we do sometimes talk about a subgenus. And with the species level, we don't talk about a superspecies, but we do sometimes talk about a subspecies. So with humans, modern humans, Homo sapiens like us, are sometimes considered to be a separate subspecies from a fossil relative that we usually call Neanderthal man. So if we have a subspecies, there's usually three names. So for instance, if we're talking about humans as being a subspecies, we would talk about our subspecies as actually Homo sapiens sapiens. Don't know why they use the same word twice. But so the last name here, so we still have the genus, the first part of the name. The second part is still the species. The third part is the subspecies. In this system of classification, if we were talking about Neanderthal man, the subspecies is Neanderthalensis. So we might see a subspecies occasionally. Notice when you see a species name, sometimes you also see a person's name after this. You might see, Homo sapiens Griffith. What does this mean? Well, that this person named Griffith discovered this species of living organism and named them. So that's one way that you can, in a sense, become famous. If you discover a new species, then your name is attached to that species, so to speak.
So all subspecies, they can interbreed?
Well, with subspecies, remember when we talked about phyletic gradualism, we said, we have groups that are become gradually more different. And with subspecies, the evidence is that in most cases, they can probably interbreed. So for instance, with humans, sometimes Neanderthal man is considered to be a subspecies. Sometimes they're just put in the same species with us. But we know that there's evidence that Neanderthal man and modern humans, as we're usually called, interbred in Europe and the Middle East. So usually subspecies can interbreed. We can't say always because unfortunately, nature doesn't cooperate and give us things that are always true. We would like things to be nice and clean cut. Nature is not so cooperative. Okay, so remember what we said earlier, if we're talking about the domain bacteria or the domain archaea, or if we're talking about eukaryotes that aren't plants, animals, fungi, other than the domain, pretty much we use the genus and species. As I say, there are some people that attempt to divide bacteria into classes and orders and families. But at least in my experience, I haven't seen too many books on bacteria that agree on the classes and orders and families. And one of the reasons, if you think about it from a modern genetic perspective, why that's true, if we look at primates, things that are related to humans, for instance, we've been able to extract DNA from fossil Neanderthal bones. A lot of the DNA in Neanderthal bones is the same as in people. If we look at some of our close relatives, like chimpanzees, there are living chimpanzees, so it's pretty easy to get DNA from them. But what we see is they still have a lot of DNA in common with people. They don't have quite as much as Neanderthals do. And if we go way back, few years ago, you may have seen a study that said that, about 98% of the DNA in a person is the same as the DNA in a chimpanzee. And a lot of people were offended by this. But keep in mind, about 50% of the DNA in a person is the same as the DNA in a banana. Bananas aren't even animals, but what do they have in common with us? They're eukarya. They have eukaryotic cells. So actually, a lot of their DNA is the same as ours. Now, the DNA that's different, that's DNA that, makes plants different from animals. But a lot of the biochemistry that goes on in a banana cell happens in a human cell too. So the DNA is the same. So when we get to bacteria, whether it's the domain bacteria or the domain archaea, the two groups that have prokaryotic cells. And when we talk about eukaryotes that aren't plants, animals, or fungi, we'll talk about the genus and species, but the other groups that we'll talk about are things that are called clades. And a clade is kind of a generic term for a group of related organisms. But a clade could potentially fit in kind of at any level of classification. We'll talk more about clades in just a moment. So we want to keep in mind when we look at these levels that if we're talking about domain, the domain has a very specific biological meaning. The three domains are based on cellular characteristics. Eukaryotic cells have a nucleus and other membrane-bound organelles: endoplasmic reticulum, Golgi apparatus, vesicles, vacuoles, mitochondria, chloroplasts, things like that. Prokaryotic cells, and that's what we find in archaea and bacteria, don't have a nucleus, they don't have membrane-bound organelles. And then if we look at cellular metabolism, the cellular metabolism of archaea is a little bit different than bacteria, a little bit different than eukarya. Turns out to have what scientists believe are more important similarities to eukarya. But as a result, once we learn the criterion, we can look at cells under a microscope, or we can do some metabolic tests on cells, and we could all agree that something is either bacteria or archaea or eukarya. That would not be controversial. If we look at organisms that have to mate with another individual to reproduce sexually, notice we're only talking about organisms that mate with another individual. Some organisms can make sperm and eggs, so they reproduce sexually, but they don't need another individual. In this case, species is defined by the ability to reproduce together. So this means that species also has a biological definition, but only in this unusual case. For all the other levels of classification, so for the genus level, the family, the class, the order, the phylum, and even at the species level, if we're talking about organisms that either reproduce asexually or reproduce sexually but don't need a mate. Biological characteristics are somewhat arbitrary. And what I mean by that, if you and I were both experts on bears, we might divide the Ursidae up into different genuses. And it wouldn't mean that one of us is right and one of us is wrong, but we would do that because we were giving more importance to different characteristics. So, you know, black bears, yeah, there's a little bit of controversy about the species between splitters and lumpers, but, you know, we pretty much agree. But exactly what is a class? Well, we have some mammals that don't do things that we associate with mammals. Has anybody ever heard of a group of mammals called monotremes? And if you've heard of monotremes, does anybody know what the two living monotremes are? They're considered to be mammals. They're a subdivision of mammals. Remember yesterday we said there were 3 subdivisions of mammals, and I think I mentioned 2 of them.
Are those the egg-laying mammals?
Yeah. Monotremes are, one of them is something called a spiny anteater, and one of them that you might have heard of, you might not have known it was a monotreme, is something called a duck-billed platypus. And a duck-billed platypus is sometimes described as an animal that looks like it was put together by a committee. And if you've ever had to be on a committee, you know, one of the things that happens on a lot of committees is you're trying to keep everybody on this committee happy. So you kind of take a little bit of everybody's ideas and you end up with kind of a mishmash of stuff that doesn't really go together very well and doesn't work. Well, if you look at a duck-billed platypus, they do have a bill kind of like a duck, and that's how they get their name. Their bill actually contains receptor cells. Now, we have receptor cells that detect light in our eyes. We have receptor cells that detect chemicals in our nose and our tongue. We have receptor cells that detect pressure in our skin. Duck-billed platypus has receptors that detect electricity. So duck-billed platypus live in muddy water. They put out electrical currents. The currents bounce back, and they can actually, in a sense, see their environment by reading the electrical reflections. Works very similar to the way that sonar or radar works. Well, okay, that's kind of strange enough, but if you look at a duck-billed platypus, they do have hair, so in that sense, they're kind of like a mammal. But their feet are webbed feet. They swim, so lots of animals that swim have webbed feet, but mammals don't generally have webbed feet. And they have a wide tail, kind of like a beaver. And as was mentioned, I think it was Nick, but they lay eggs. They don't form a placenta. So monotremes were one of the first types of mammals to evolve. And although there's only 2 species alive today, the duck-billed platypus and the spiny anteater, if we go back and look at fossils, they were once much more common. So if we look at them, the point I was trying to make is, okay, those are in the group Mammalia, but they're kind of borderline mammals. There are some dinosaurs that are considered to be reptiles. Dinosaurs are grouped with the reptiles, but there are some dinosaurs that actually have feathers. So are they dinosaurs or are they very early birds? This is what I mean when I say that, you know, our criteria for these levels, except for the domain and the species, is kind of arbitrary. We could all be specialists in studying mammals, and we might decide to divide mammals up into different orders. And it wouldn't necessarily mean someone is wrong. And although in some ways, when we talk about dividing living things into categories, it's kind of boring, I'll admit. When you read the professional journals in biology, the professional journals about taxonomy and classifying living organisms have some of the most heated arguments of any professional journal. And it might seem contradictory, but it actually makes sense, because this is why. When you divide living organisms up into categories, how do you divide them up? Well, you use some of their characteristics. You say, well, this characteristic separates mammals from their reptile ancestors. But what characteristics you use to classify things depends on your point of view about what's important for living organisms. So when people are dividing things into categories and they get into an argument about what's a mammal and what's a reptile, they're not really arguing about mammals and reptiles. They're arguing about what characteristics are important in biology. And so as a result, as I say, the arguments are really obnoxious. Now, after Darwin's book was published in 1859, remember what I said, his book was kind of the equivalent at the time of a bestseller. They didn't use that term back then, but biologists pretty much agreed that the classification of organisms had to reflect their evolutionary relationships. Some classification schemes before Darwin put animals like bats, which are mammals, and birds, which are birds, and insects that fly in the same group because they all fly. And animals like sharks, which are fish, and dolphins, which are mammals, were put in the same group because they both swim in the water. And after Darwin published his book, biologists decided that the way we divide things up into categories should reflect their evolutionary relationship. The categories that we have should be organisms that are closely related. So when we talk about bears, bears should be closely related to each other. They should be more distantly related to other carnivores like the cats and the dogs. And so biologists began classifying things, and this type of classification was sometimes just called evolutionary classification. The problem was what I mentioned a moment ago when I was talking about why you might have some conflict in terms of which group an organism fits in. The problem was, this was, and this should be being, not begin, I typed that wrong. The problem was this was very subjective. And it's really subjective when we start studying organisms that are quite different from people. If we look at mammals, I think for most of us, we could probably agree on what characteristics are important for one group of mammals versus another group. But what happens when you're looking at a group of beetles that are almost microscopic? Is eyesight really important to these organisms? Is the length of the leg important? It can be very difficult to classify organisms that are quite different from us. And what about plants and fungi? How do we divide them up into meaningful categories? Because, the problem is when we look at other organisms, we tend to look at them from our perspective. Well, wait, we really rely on our eyesight. Yet it's very well established that if we look at most mammals don't rely on their sense of vision. Does anybody know what most mammals rely on to get information about the world? I mean, they do have eyes, they can look at the world, but if you look at most animals and you look at their face, if you've got, a dog or a cat at home, or, if you think about a horse or a fox, what sensory organ is at the front of their body?
The nose?
The nose. Most animals get information about their environment by smell. So we're a little bit different. Now, the reason we're different, we're pretty sure, is because we evolved from other primates. And most primates live in trees. So one of the advantages of having two eyes that aren't really very well separated by your nose like we have is that allows you to get pretty good depth perception. You know, if you look at your dog, one of the things we know from studying dogs, you know, dogs have kind of a big nose, and the nose kind of separates their right eye and left eye. Their right eye and left eye can look at different things at the same time. Now, their brains have to be wired a little bit different. If you would try to do that, your brain would not be very happy. But that means that they have a lot of peripheral vision. But since both eyes aren't looking at the same thing, they don't have great depth perception. So if you have a dog or a cat, one of the things you may have noticed, a lot of times they jump for their toy or whatever, and they miss. Or if you have a cat, and cats like to jump up on things, a lot of times they jump and they don't quite make it, or they overshoot their mark. So their depth perception isn't great. But since we evolved from primates, primates live in trees. Well, wait a minute. If you're sitting on a branch and you think the next branch is 3 feet away and it's 6 feet away, it has a much more drastic consequence if you jump for a 3-foot branch and you miss it. Because if you're up in the air 30 feet, that might be the last jump you miscalculate. So the problem with looking at other organisms is it's very subjective. So in the 1950s, a professor at the University of Kansas, Robert Sokol, who was strongly influenced by Sewell Wright, who's actually was at the University of Chicago here, he started work on a new type of taxonomy. And eventually this became what was called numerical taxonomy. And one of the things that helped numerical taxonomy advance at this point was that computers were just starting to come into use. Now, keep in mind, computers were not what we think of today. You may have read, but if you haven't, you know, a computer that is as powerful as, you know, your Surface or your Mac at home that you have now, that type of computing power would take up a whole room filled with different components. And in fact, if you have a smartphone now, your smartphone is more powerful than the computers that the Defense Department used during the 1960s to protect us against nuclear war. So at this time, computers weren't great, but computers could do repetitive work and they could do it very fast. And what Sokol said was, here's what we need to do. We're going to measure as many things as we can when we study organisms. We're going to measure the length of their toes. We're going to look at how many toes they have. We're going to look at the length of their nails. We're going to look at the width of their nails. We're going to look at the width of the toes. We're going to look at the depth, how high they are from top to bottom. You know, we're going to look at the color of the toes. We're going to look at everything we can. Things that we can't measure, like the color, you know, we might be able to compare them to a color wheel, like if you were painting, but things that we can't measure, we're gonna assign a numeric value. You know, maybe 1 is white, 2 is pink, 3 is yellow, 4 is orange, whatever. So we're going to measure as many things as we can. We're not going to worry about whether we think they're important or not. All characteristics are going to be treated as though they're equally important. And then what we do is we make a big grid, and we see, you know, if we look at black bears versus grizzly bears versus Kodiak bears versus polar bears versus sun bears, we're going to have this grid of all these characteristics that we've measured. Now, Sokol said you should measure at least 50 different things, and really you should measure more than 100 if you want to be very accurate. Well, once we decide what all these measurements are in all these different groups that we're trying to sort out, now we use the computer to figure out which characteristics these groups have in common. And the more characteristics the groups share, the more closely related they are. The fewer characteristics they share, the less closely related they are. Now, notice this is a good way of getting around the problem of looking at characteristics that we think are important. We're looking at as many characteristics as we can. We're not really worrying about whether we think they're important or not. So this started to solve that problem. But numerical taxonomy had some of its own difficulties. It wasn't perfect. So we ended up getting things called simple matching coefficients that told us how closely related groups were. And the simple matching coefficient was essentially like a frequency, was a number between 0 and 1. Organisms that didn't have anything in common, their simple matching coefficient would be 0. If they had everything in common, the simple matching coefficient would be 1. Maybe because the mathematics was pretty complex, numerical taxonomy never really replaced evolutionary taxonomy very fully. There were a few people who did numerical taxonomy, but for the most part, people stuck with evolutionary taxonomy the same way it had been practiced since shortly after Darwin's book. Around the same time that Sokal was doing his work, there was a German named Willy Henning who was working on another type of classification called cladistics. And cladistics is now accepted worldwide as the best way to study evolutionary relationships and classify organisms. Now, cladistics still has some issues, which we'll talk about, but Henning would have become famous sooner, but he worked in Germany. All his original papers and books were published in German, and they didn't start to get translated into English until about 13 or 14 years after he wrote them. And, you know, if we would go back to the 1700s or 1800s, most scientific papers were written in either German or French. But by the 1960s, English had become the dominant language in science. And so the fact that all his papers were written in German meant they were kind of ignored. And cladistics has 3 basic ideas. Remember I said earlier that we could call some of our taxonomic levels clades if we wanted to. And in cladistics, all the organisms that we put in a clade, doesn't matter how many there are, whether there's 2 or 1,002, all the organisms in a clade are related. How are they related? Because they've descended from a common ancestor. So it's that process of divergent evolution being repeated, perhaps leading to adaptive radiation, but everybody has to come from the same ancestor to be in a clade. If they don't come from the same common ancestor, they shouldn't be in that group. Kind of like you might trace your family back to your great-great-grandfather. And so your family includes everyone who is descended from your great-great-grandfather, but it doesn't include people who haven't descended from him. And this idea wasn't really argued by the people who were practicing evolutionary taxonomy, because that was what they were going for too. Now, the second thing that cladistics said was the characteristics of living things change over time. Well, again, nobody really debated this. This is really what the, you know, if you wanted to summarize things, this is a good summary of evolution. So people didn't argue about that. Now, their third idea was controversial. They said you get new species being formed by what they called bifurcations, splitting into 2 of an existing species. And the existing species then becomes the common ancestor of these 2 new species. So let's look at an example of, you could think of this as a family tree. In cladistics, we usually call this a cladogram. Now, remember when we talked about our geologic time scale, we said the oldest time is down on the bottom. And the most recent time is up at the top. So if we go down to the bottom where we have this single line sticking down, this is the oldest organism in this group. And keep in mind, it doesn't represent a single organism. It represents a population, and could be multiple populations because it's a species. And remember, our definition of species is it's a group of populations. So this vertical line where they have this gold box at the top, this organism is the common ancestor of species A, B, and C. Why? Because species A, B, and C are all formed by branches that come off of this common ancestor. Now notice that A is the first species that branches off. And then over here, if we look at this short segment before we branch into B and C, this short segment is the common ancestor of just B and C. It's not the common ancestor of A, notice, because A has already branched off over on the left side. And really, if you wanted, you could extend this green box all the way down to the bottom of this vertical line. And the same thing with this gold box. And then if we're looking at any of our individual groups, this vertical line represents the ancestors of species C. And similarly, we could have a box over here on the line underneath B. This would represent the ancestors of species B. Or we could have a box on this line underneath A. That would represent the ancestors of species A. Now, here's what we have to think about. A lot of times on cladograms, they don't really tell us, but if you think of this as a graph, just like our geological time scale, the y-axis is time. So again, the most distant time is down at the bottom. The most recent time is up at the top. So if we look at our cladogram, what does it really tell us? Well, it tells us how closely organisms are related. And the basic rule is a simple one. The more recently two organisms have branched away from each other, the more recently they've separated, the more closely related they are. So if we look at A, B, and C on this cladogram, A branched off first. So A is least closely related to B and C. Why? Because it branched off first. B and C are the two species on this cladogram that are most closely related. Why? Because, think about the time scale. They've separated from each other. They've branched off from each other most recently. So the more recently two organisms have branched, the more closely related they are to each other. So we want to keep that in mind because we're going to look at some cladograms. And when we start looking at the cladograms, one of the things that we'll see is that almost all the questions, the answers are going to have to do with, did they branch off from each other more recently or longer ago? That is almost always going to be our answer.
I have a question.
Sure.
So looking at that diagram, it seems like it's assumed that each common ancestor becomes extinct.
That is kind of an assumption, and that's actually been one of the arguments that people have against cladistics. If we focus on this common ancestor of A, B, and C, we have some kind of a speciation event where we see this goalpost split. And in cladistics, we assume that, okay, one branch of the family forms species A, and the other branch forms the thing that eventually becomes B and C. But one of the things we see in the fossil record is sometimes we have an ancestral species and a small population splits off, we get a new species, and the ancestral species is still there. So yeah, that's a good observation. That is one of the complaints. Another complaint is that cladistics says it's always a bifurcation. It's always splitting into two groups. You know, what if you're talking about a group like our silver sword plants that we mentioned a little while ago, and you have different plants land on different islands at the same time? And, you know, why can't we have a common ancestor split into 4 groups at the same time? Why does it have to be 2? But yes, you are correct. That is an argument against cladeism, and that's why, as I said earlier, this isn't a perfect system. You know, it's not as nice as we would like it to be, but it's still considered to be better than numerical taxonomy or just classical evolutionary taxonomy. So let's look at a cladogram of primate evolution. So here we have primate evolution, and notice this is kind of a magnified version of our geological column. This is the part that was in green when we looked before, the Cenozoic era, the age of mammals. So if we would go down to the very bottom, you know, here's 60 million years ago, this line here. So a little bit before that, we have the time when dinosaurs became extinct. So notice that primates were around before dinosaurs became extinct. And if we look, the first group of primates that branches off is a group called the lemurs and the lorises and the patos. And especially lemurs, you might have seen if you've gone to the zoo. But if you look at all of these, they're not what most of us think of when we think of primates. Most of us, when we think of primates, we think of monkeys or apes. And lemurs and lorises and patos kind of look more like squirrels maybe than primates. They don't have the big bushy tail in general, but that's kind of more the appearance they have. Then we have another group that branches off, the tarsiers. And they tend to have very long, stringy tails, and they usually have very large eyes for their body size. But again, their body size, they're pretty small compared to what we think of when we think of primates. They have the large eyes. Remember when we were talking about vision before being important in primates? Tarsiers are some of the few primates that are partly nocturnal. Because primates rely on vision so much, most primates are active during the day when there's more sunlight available. But one way to gather more light is to have bigger eyes. Then if we look, the New World monkeys branch off. And the New World monkeys are monkeys that are found in South America and Central America. They do show a pretty good picture. You know, one of the ways you can impress your friends when you go to the zoo, this picture shows a New World monkey hanging by one arm from a branch and also by its tail. New World monkeys have prehensile tails. They can hold on to things with their tails. Old World monkeys don't have prehensile tails. Old World monkeys, the tails are used for balance, but they can't really hold on to a branch or grasp something with their tail. Then if we look, the Old World monkeys branch off, and the Old World monkeys are found in Africa, Asia, the Middle East, Europe. Keep in mind, you know, this is the Europeans' point of view. The Old World is what they were familiar with. The New World, the Americas, you know, were recently discovered. And then in this branch of the family that goes off to the right, kind of opposite Old World monkeys, what we see is what are sometimes called the apes. And we have a couple of main groups. We have the gibbons, which are sometimes considered to be apes, sometimes not. And gibbons are brachiators. Gibbons swing through the jungle. If you've ever seen the Tarzan movies, when Tarzan swings through the jungle, that's how gibbons travel. And then we have orangutans, gorillas, and then notice on this chart, the group that's most closely related is chimpanzees and humans. Now let's look at orangutans for a moment. And suppose I asked you which group of primates are orangutans most closely related to? Well, remember what we're looking for when we look at how closely related organisms are is how recently have they separated? So if we look almost directly under the word gorilla, if we look where this vertical line comes up, this is where orangutans separate from the other great apes. Well, notice orangutans separate. The common ancestor of gorillas and chimpanzees is in here. The common ancestor of chimpanzees and humans is over here. Notice that orangutans separated from the primates that led to gorillas and chimpanzees and humans at the same time. So the primates that orangutans are most closely related to are gorillas and chimpanzees and humans equally. They're not more closely related to one of those groups than the others. Even though it might look like they're more closely related to gorillas because the word gorillas is written closer to orangutans, that's not the case. Let's leave this for a minute, and I want to go to screen sharing with our whiteboard so that we can see what I mean. So let's look at a cladogram just with 3 organisms. Gorillas, and chimps, and humans. So chimpanzees and humans are more closely related to one another than they are to gorillas. So we could represent that relationship in this fashion. But notice we could also represent it in this fashion, and this says exactly the same thing. Notice I flip-flopped the H for humans and the C for chimpanzees. Doesn't matter which I write on the left and which I write on the right in this group, humans and chimpanzees. They're both equally related to gorillas because in both cases, if we look at this spot, it's sometimes called a node. If we look at where gorillas separated from the common ancestor of chimps and humans, it's separated from the common ancestor of chimps and humans at the same point in time. So gorillas are just as closely related to chimpanzees as they are to humans. Doesn't matter which one we write on the right or left. And in fact, we could write the same cladogram like this if we wanted. This says the same thing. What we're interested in the cladogram is the branching pattern. So notice in all 3 of these, what happens? The gorilla side of the family separates from the human and chimpanzee side of the family first, and then later humans and chimpanzees separate. So all 3 of us give that same information. So when we're looking at a cladogram, we don't want to get focused on, is humans written on the right or the left? When we're talking about who they're related to, it's when did they branch from someone? Any questions about that idea? Okay, we're going to go back to our other diagram. So on this diagram, chimps and humans are the most closely related. Now notice this diagram is grossly oversimplified in the sense that there are 2 species of chimpanzees that are extant, which means they're not extinct, they're living organisms now. And there are numerous species of humans. There's only one that's extant, us, Homo sapiens. All the other relatives of humans are extinct. There are 2 species of gorillas. There are lots of species of old world monkeys, new world monkeys. So we could potentially have a lot of smaller branches in here. But on this diagram, The two species that are shown that are most closely related to one another are chimpanzees and humans. Now, let me ask you another question. Suppose you on this diagram, I ask you which primates are least closely related to the other primates. What would you tell me if I asked you which primates on this diagram are least closely related to the others? So when you hear least closely related, what you want to think about is who branched off from the other primates first. So our answer here would be these lemurs and lorises and patos. So just like humans and chimps are most closely related because they've separated most recently, lemurs, lorises, and patos are least closely related because they separated longest ago. Now, suppose I ask you a question about Old World monkeys. Are Old World monkeys more closely related to orangutans, or are they more closely related to New World monkeys? So Old World monkeys, are they more closely related to orangutans or to New World monkeys? So think
New
about what I've
World,
been, go ahead.
New World monkeys?
Well, remember, all we care about is the split. So notice here, since Old World monkeys split from New World monkeys longer ago than they split from orangutans, the group they split from most recently, orangutans, that's the group they're more closely related to. Even though there's a couple of extra branches on this side, that doesn't matter. So sometimes that can be a little bit deceptive. So that's very tempting to say New World monkeys, but all we're looking at is how recently did the two groups separate? The more recently they separated, the more closely related they are. Now, if we look at how we get this cladogram, how we put things into groups, one of the main differences between cladistics and the other types of classification we talked about is how cladistics thinks about similarities between groups of organisms. Cladistics says there's really three types of similarities that we see between organisms. We see homologous characteristics. Homologous characteristics are characteristics that two clades have in common because of convergent evolution. So remember yesterday when we talked a little bit about ichthyosaurs, reptiles, and dolphins, mammals, and sharks, fish. So homologous characteristics are characteristics that things have in common, not because they're related, but because they live in the same environment and they've gone through convergent evolution. So this doesn't matter when we're trying to figure out how closely related things are. You know, would be like if I was looking at people trying to figure out who in a group of people was related. If I'm looking at something, if you inherited blue eyes from your parent and someone else inherited blue eyes from their parents, it doesn't mean that the two of you are related. You've got those from different ancestors. Another characteristic that isn't important, according to cladistics, is shared ancestral characteristics. And these are things that are found in two clades. And keep in mind, when we say two clades here, we go back to our diagram, we're thinking about two clades like chimpanzees and humans. And so we're going to think about chimpanzees and humans and their common ancestor. So these are characteristics that are found in two clades of organisms, and they're also found in the common ancestor. So this can help tell us that these two groups are related, but it doesn't really give us any help in telling us that these two groups are different from each other. So for instance, imagine looking at humans and chimpanzees and saying, well, humans and chimpanzees both have 4 limbs. They have 2 hind limbs and 2 forelimbs. So that's a similarity. Well, yeah, but everything in the group primates has 4 limbs. And in fact, all mammals have 4 limbs. And in fact, amphibians, reptiles, birds, and mammals are called tetrapods. They all have 4 limbs. Even snakes, if we look at the embryo of a snake, snakes start to develop legs and arms, and then those parts of the body get reabsorbed during embryonic development. And this is because snakes evolved from ancestors that had legs and arms. So if we're trying to separate organisms, the only thing that cladistics says is useful is something that it calls shared derived characteristics. Things that have evolved after the split. That's what we can use to separate organisms into categories. So a lot of times when we draw a cladogram, we actually list the characteristics that evolved. So for instance, down here, kind of in the middle of this slanting line in the lower part, this is where our common ancestor of all these organisms is. And this common ancestor at some point evolved vertebrae, a backbone. And after it evolved a backbone, then one group of organisms, they show sharks, but notice there are some other things that are related to sharks called rays and some other organisms, hagfish. That branch separated from the rest of the vertebrates. And then a bony skeleton developed. And after the bony skeleton developed, a group of fish that we call the ray fin fish, and this is really fish that you're familiar with, things like minnows or bass or trout or salmon. Almost all the fish we think of other than sharks are ray fin fish. That branch separated from the vertebrates. And at that point, this little line in here that was the common ancestor of all these other organisms, some of the bones in those fins lengthened, got thicker, and eventually became useful for walking. And so the common ancestor developed 4 limbs. And 4 limbs are found in amphibians and reptiles and birds and mammals. And then we have something called the amniotic egg and eggs with shells. So a lot of times we can put the evolution of some characteristic in here, and the evolution of this characteristic is partly what separates groups. If we look at crocodiles and birds both have eggs with shells. If we look at mammals, we have an amniotic egg, and that's the same type of egg that we find in crocodiles and birds, but our eggs don't have shells. Well, except for a few of those weird duck-billed platypus and monotremes, animals in the same group. So when we look at a cladogram, a lot of times we'll have different characteristics listed. And these are the shared derived characteristics that are separating one group from another group. Now, keep in mind, despite the fact that I've said cladistics is the best method that we have for studying classification of living organisms, depending on which characteristics you look at, you might get different results. And this is using cladistics as an example, but the same thing applies to evolutionary classification or to numerical classification. And a good example is turtles. If we look at morphology, and morphology is always anatomy, it's the parts of the body. If we look at morphology of turtles, they appear to be the first group to split off from all the other reptiles. Because you know, that shell on a turtle is pretty weird. And actually, when you look at turtles, the shell grows out of their ribs. So not only is the shell unusual, but think about that. Your ribs are inside your body. In a turtle, the ribs grow outside the body, and then, you know, ribs are kind of thin. Some tissue grows in between the ribs to fill out the shell. But on the other hand, if you look at DNA, lizards seem to be the first group to split off from all the other reptiles. And turtles are pretty closely related to crocodilians, crocodiles, caimans, things like that, alligators, and birds. So which is right? Neither one is. Or maybe a better way for me to say it would be to say, we can't tell which is right. We need more information. So just because we have a good way of studying things, we don't want to develop tunnel vision and think that now we know how all these organisms are related to one another, because we don't. You know, what we have now is our best guess. The best thing I can tell you about classification is a story that I sometimes tell my classes about a friend I have who made a graduation speech at a medical school. It's probably about 10 or 12 years ago now. So it was the you know, these doctors had been in med school. They were graduating with their MDs. And this guy got up and he told all the doctors, he said, well, I have some good news and some bad news to tell you about your medical education that you've gotten the last few years. And he said, the good news is half of everything we've taught you is wrong. And all these new doctors gasped and kind of shrieked, and he let everything die down. And he said, the good news is that half of everything we've taught you is wrong. He said, the bad news is we don't know which half yet. And that's kind of a good way to think about classification. The classification scheme that we have right now that we'll talk about as we go through the semester is the best one that we have with our current knowledge. But as we discover more fossils, as we learn more about the DNA, because keep in mind, if you look at a human, 98% of the DNA is not the instructions to make proteins. And we don't know what a lot of that 98% does. When we discover that, that may lead us to figure out some relationships that right now we don't know. Because right now, when we try to see if organisms are related by their DNA, a lot of what we look at is the proteins they make and the DNA, which is the instructions for those proteins. So, you know, that was one of the reasons why, and keep in mind, we're going to get to it in about 2 or 3 weeks. But if you don't have a copy of this book, the textbook that we're using, Campbell Focus on Biology, you want to see if you can get a copy of the chapter on eukaryotic diversity. Because the chapter on eukaryotic diversity is different in every book. And it's different in our book than in other books, or that you're going to find if you do a Google search. So you want to have a copy of our textbook to answer those questions. Are there any questions about what we've talked about today? Okay, if not, I'm going to let you know. Did anybody try to go to YouTube and see any of our videos? If you haven't, let me know if you go there, if the videos don't come up or anything, so that I can see. I think I have the videos that we've done so far so that they're visible. But let me know if something doesn't work and I'll see if I need to adjust something. Otherwise, if there are no questions, I'll let you go. Tomorrow, we're going to get a few problems that will be due early next week. And we're just about finished talking about evolution. So at some point, I will have a large set of questions that I'll give you a whole week to work on. That will be kind of our summary. If we were in class now on campus, it would probably be the type of questions that are on our test. But since we're not there, I do like a lot of regular assignments, and you just have a couple of days to work on them. So it's more questions, so you will have about a week to work on those questions. So if you don't have any questions, go ahead. Go ahead, Samuel.
So I went through the YouTube with your name, but like I think is it that it's more difficult like posting it on YouTube? Is it like it takes more time because you're saying
It
something
takes more
about
time. Sometimes it takes an hour or two to upload the video. You know, if you think about if you've ever watched other videos on YouTube, most of them are pretty short.
right?
And I had to get a special license to post videos longer than 15
I
minutes.
know.
And then for days like today, when we're even longer, I have to get special permission to post something longer than an hour.
Oh, okay.
Yeah. Yeah. So sometime soon, hopefully later today, I should have this one up and the video from yesterday, both.
Okay. Thank you very much.
You're welcome. I think all the other ones should be up. I know there's not one from Monday this week because I forgot to record.
Yeah, I should record.
Yeah. So like I say, if you ever notice I'm not recording, remind me so we can get started.
Right. Thank you very much.
You're welcome. Enjoy the rest of the day.
Yeah. Thank you. Bye.
Bye-bye.
Dr.
I'm
Griffith.
sorry.
Dr. Griffith.
Go ahead.
You know the monohybrid cross assignments. Do you have to turn those in?
Not yet. I'm going to give you one tomorrow that you will have to turn in.
Okay. Okay, thank you.
So one example of a Hardy-Weinberg and one example of a natural selection, both.
Okay, thank you.
You're welcome.
So those chapter questions that I do, you're going to give those tomorrow as well as the lab, or is that lab in our book?
The lab is separate, so we'll talk about that, but that's not going to be due for about a week and a half.
Okay, thank you.
You're welcome.
Have a good day.
You too.
You said the lab assignments are based on questions in the book, in the textbook?
Well, I pretty much all the questions that we have, I think you'll find the answers either in the book or in the notes or in the Zoom videos.
But
I
you were
know
saying
last
that,
semester there was one student who went back and looked at some of the Zoom videos again and looked at some of the notes and told me that they could find a lot of the answers in there. They're not always in the same order they are when I give you a set of questions. So I would recommend reading all the questions first if you are going to go through the notes or review the Zoom video, because otherwise you'll kind of have to backtrack a lot.
you said that some of the questions that you were going to give us, we're going to have to look inside the book to answer them.
Right.
Okay. Okay, thank you.
So that's why I was saying the chapter on eukaryotic diversity, you want to make sure that you have the book for that, because that's one place where the questions definitely come from our book.
Okay. Okay, thank you.
You're welcome.
Mr. Griffith, the book I have, the edition I have it's not really the updated edition, like last edition.
Is it the second or the first edition? I think the one we have is the third.
The second. I have the second.
I'll check because I have a copy of both of them. I think the second edition is okay. I think the first edition, though, they made some major changes to that part of the book before they did the second edition, but I'll double check for you.
All right. Thank you.
You're welcome.
Bye.
Bye-bye.
Take care.
You too, Nick.
Okay, Professor.
Mm-hmm.
I know I'm totally behind because I didn't even know we had assignments to do. So where exactly can I find these?
Well, the first assignment was a note card, and that should be in the week 1 module. The second assignment was a genetics problem, and that's in the week 2 module. And then tomorrow I'm going to add something to this is the week 3 module that will be due next week.
Okay, all right, thank you.
Yeah, so go back, double check. If you don't see it, send me an email and I'll double check. I think it's still visible, but I'm not positive.
Okay, thank you. Have a
You're
good
welcome.
day.
Okay, Mr. Griffith, for the lab homework, do we have to get the lab book, then do the homework in it?
No,
Okay.
for that, I will, like if you remember the pages a couple days ago that we did with like the little pictures of the rabbits and the deer and stuff,
Yeah.
that's actually from the lab book.
Okay.
So if there's something I want you to do that's out of the lab book, I'll send you those pages.
Oh, okay. Thank you.
You're welcome.
All right. Bye.
Bye-bye.